Got it. Okay. And I'll be focusing on brain organoids with a very provocative question, which is, where does cognition begin? And the idea is that to create a useful model where we can study sophisticated networks that might give rise to cognition, self-awareness, and any behavior from codified by the human brain. And this is quite hard because we don't even know if a newborn baby is actually self-aware or has any cognition. And we are even trying to recapitulate that in a dish. So there is many reasons to believe that this might not even be possible. So we use this technology of a mini brain in a dish, which has several limitations. Most of the protocols, they're dealing with immature neurons. These structures are not vascularized. Not all cell types are represented. We don't even know if we're growing these cells in the right condition. So what my lab does is take time to optimize these protocols, optimize how we grow them. And long story short, so it has been like 3 years that we are optimizing a novel protocol where we can keep these brain organoids in the right conditions and optimized conditions that they can live and survive for months, if not years. And there is several things that is important to highlight. One aspect of that is when you make these organoids, you don't make a single one, but you make several of them. Here is a postdoc in the lab growing them, and you can see that, first of all, they are about 0.5 centimeter in diameter, so you can see it the naked eye. They can grow up to 2 years, and you can grow hundreds, if not thousands of them. But the really big questions that we have is that can this be a novel way to understand sophisticated neuronal networks? And this is something that wasn't addressed in previous protocols. And we thought that we should be doing that by comparing with previous work. So what I have here is just the mean firing rate. And this is just a measure of overall brain power. And the work that I'm showing here is all the work done with 2D cultures. This is our regular traditional cultures. So most of the work is below 5 hertz as one of the measurements. And we are quite far away from what we consider like a sophisticated network coming from a mouse brain or even a monkey brain. So even brain organoids can be kept for several weeks in culture. But by doing that, you don't see an increase in the sophistication of the network. They are still below the 5 hertz. So I think the biggest surprise is that when we start growing or using this protocol over time, we see that not only we can speed up the maturation of those neurons, but we can reach to a level that is close to a functional monkey or mouse brain. So that opens different possibilities, including the study of neural oscillations that you can only achieve if you have a high-power brain activity. So these neural oscillations are important because you can record through the skull. So these are the waves that you see if you put like an EEG cap like this one. And they basically are correlated to virtually any behavior in humans. But to understand what are the age of the organoid, we use these EEG or electroencephalogram features from preterm babies to compare with our brain organoid. And we use a machine learning to do that. So we use unsupervised learning algorithm to teach the machine to tell the age based on the EEG from several preterm babies. And after that, I mean, we start feeding them with our brain organoid electrical activity. And what the machines tell us is that there is a really good prediction after 25 weeks, but really bad prediction before that. And the reason why is that human babies do not survive when it is below 25 weeks, so we cannot like teach the machine to predict the age. But after that, I mean, we have a really good prediction. And what we learn is that our brain organoids, if captured properly in culture, they will mimic something that resembles a postnatal human brain. So we can reach that level of oscillations. So now that we have these oscillations in a dish, I mean, we start asking, can we use these brain organoids to test drugs for conditions where these oscillations are affected, for example, in autism or in schizophrenia? So this is a company, a startup that I'm a co-founder, that we are using these brain oscillations as a readout for these drug screening platforms. So another perspective of this oscillation is that they kind of reach a plateau after 9 months. And this is what we expect for the human brain. I mean, when we have like a baby that was just born, it's still like an immature brain. And the only way for this brain to mature is to have input or output. So the way we decided to do that was to plug these brain organoids into robots. So we are teaching the robots to interpret the oscillations that are coming from these brain organoids and explore the world. So as they explore the world, we are creating like a neurofeedback using optogenetics or posing them with neurotransmitters so they can start learning something. So what we have here is a video, I think you can play the video, of the initial attempt to make a robot learning using these brain organoids. So what you have here in the video, if you can just play it, it is the organoids are actually not inside the robot, they are in the lab, but they are sending signals to a computer who sends the codification for the movement to the computer, to the robot wireless. And it takes about 10 seconds to do that, so we are trying to reduce the speed of that. And again, I mean, if you're not impressed with this movement of the robots, you should, because that's exactly what a learning network is trying to do. It's trying to optimize and explore the environment. So we hope that this would be like another learning paradigm to explore drugs for autism or schizophrenia, for example. So I'll if you can go back to the slides, I'll finish with one more potential applications because we talk about translational opportunities using these brain organoids. But there are more basic sciences that can be done. Let me move to my last slide, which is something that will be discussed here in these sessions, the use of genome editing to learn more about how these sophisticated networks emerge. We've been editing these brain organoids. One more. In different ways. First, we are using CRISPRs to knock down important genes that might be important for the emergence of the networks. But we also are taking an evolutionary approach. So this is just like an article from today from Science magazine showing our attempts to Neanderthalize a human brain organoid, so we can go back in time and create organoids that have DNA material from the Neanderthals. So by doing that, we can understand how the Neanderthal mind or brain complexes start to diverge from the modern humans. And what we find is super interesting, but I'll keep that as a teaser for another talk. So I'll stop here. And I'll move to the second speaker, which is Matthias, that we will be talking about organoids in a different tissue, the GI tract.
Thank you very much. So I would like to give you perhaps a bit more general perspective on organoids. I would like to put them in context with existing in vitro systems as well as organisms or animals to see what we can really do with organoids at the moment. So I think organoids can be defined as miniature tissues that capture key features of real tissues in terms of their architecture, the cellular composition, and to some extent the function. And I think a beautiful example of an organoid is that of the gut. And this is work that has been pioneered by Hans Klever's lab in the Netherlands. Here you can see basically in vivo, our gut is a meter-long tube that's lined with a layer of epithelial tissue, which has a very characteristic topography with these long protrusions that stand out here that are called the villi, and these little cavities that are called the crypts. And our stem cells in the gut that have a really tremendous turnover because this tissue is used so much, the cells are dividing very rapidly and generate specialized cell types constantly in these tissues. So the stem cells are basically located here in the crypts. They start to differentiate and specialize and generate the specialized cell types that are responsible for the absorptive and secretory function of the tissue that are located up here in the villi. So if you remove the stem cells from these tissues and you put them in a 3D environment and you give them the right cocktail of factors, something very magic happens in that the stem cells start to divide and give rise to these little cystic structures, like small epithelial tissues that further differentiate and then give rise to these structures that mimic remarkably well the crypts. So they also have stem cells in them, and these stem cells give rise to differentiated cells that retain the key functions of the tissue. So this is what we can call a mini-gut. And I think it's important to distinguish this advance of organoid biology and technology from traditional in vitro culture. And I think we can perhaps distinguish three main aspects here. So traditionally, we grow cells in culture from one type. We can combine multiple cells, but very often we grow them as a single cell type and in classical cell culture dishes. In organoids, multiple cell types coexist, and very often we have stem cells that live together with specialized cell types in the same three-dimensional structure. This is what makes them much more in vivo-like than a traditional cell culture setting. And so the architecture of the tissue, this is really capturing some of the complexity of the tissue also in terms of its makeup. It's much more physiological in an organoid system compared to a traditional in vitro setting. And as a consequence of these aspects here, we can capture more physiological functions in vitro compared to traditional cell cultures. And I think that's the reason why organoids, and we hope that's going to happen, can really fill this large gap between in vitro experiments that we're doing in the lab and the organism, an animal, doing in vivo experiments. This is really the hope that we can bridge this tremendous gap here. I think it's important to realize that organoids can be derived from multiple sources. So we can isolate adult stem cells from tissue and basically generate organoids from these cells. But we can also derive organoids from cells that are called pluripotent stem cells, which can either be embryonic stem cells or induced pluripotent stem cells. And we also have the possibility now to derive organoids from sort of cells that are in between these stages of the adult and the embryo that are called fetal progenitor cells. This is important because the organoids that are generated depend on the type of cell of origin in terms of their maturity or developmental stage. So if we derive organoids from pluripotent stem cells, we can either generate embryonic-like organoids, or we can derive more specialized organ-like structures that are sort of not reaching the adult level at the moment. They're sort of more in a fetal stage, I would say. However, adult stem cells, when we generate organoids from those, they probably are closer to the real adult tissues in terms of their functionality. And we also have the possibility to use cancer stem progenitor cells to derive structures that could be called cancer organoids or tumoroids. So the cell source plays an important role. And what is now the potential of organoids? I think, as we have seen just before, organoids represent a very, very important experimental tool for the biologists. This is already very clearly shown. There is a huge hope that we can use organoids for diagnostics, drug discovery, ultimately cell therapy. And we have important proof of concept studies that have already been published on multiple organoid systems that suggest that this will become a reality and will be routinely done in the future. I think it's important to point out that organoids give us a unique opportunity to study human biology and human diseases in a dish, where we don't have a possibility to work on an organism. We can't do real experiments on human embryos. So this is really, I think, perhaps the most exciting aspect of organoid technology. I think what we are seeing now is also that there is quite a lot of hype that surrounds organoids because of the expectations that we have and because there is so much at stake with these systems. And I think we should not be fooling ourselves and think that we can really generate real organs at the moment. We're not really there. And I would also say that in general, of course, what we expect is that organoids will reduce animal experimentation, but they cannot replace completely animals that are used for biological studies. So I think this is something important to keep in mind. Now, I think there is a tremendous opportunity here to improve organoid technology further. And in this context, I think engineering approaches can play an important role to solve some of the challenges that we are now encountering with organoids. The first one, I've shown here a list of issues that I think need to be addressed in the future. One clearly is reproducibility. Our organoids at the moment show quite a bit of a heterogeneity in terms of size and shape, etc. This can be improved by controlling the culture conditions even better. We have organoids that are not really physiological in terms of size, shape. They don't reach a lifetime that we would like to have in terms of deriving really mature adult tissues. They also lack complexity. You know, if you think about the real organ, and go back to the intestine as a model system, here we just represent the outermost layer of the organ, the epithelium. We don't have blood system, we don't have nerves, we don't have immune components, etc., etc. So there is a lot of things still lacking in terms of complexity that will need to be engineered back into a system in order to capture true organ-level physiology. And in that context, I think engineering has made a lot of advances over the last years, and there's a whole field that is called organs on chips, where scientists and engineers are trying to recreate physiological functions of organs using very clever microtechnology. And in this context, for instance, there have been studies from Boston showing that you can mimic breathing motions of the lung by engineering micro devices in which cells from the lung can be cultured and thereby capture some functionality of the liver. And so I think bridging or merging together microtechnology and engineering with organoid biology is a very exciting avenue in the future. This is what I wanted to talk about. So with that, I hand over to Rossi Gage or Shuibing Chen. Yeah.
Okay, morning everyone. I'm Shuibing Chen from Weill Cornell. So here I will give you some introduction how we use CRISPR technology and combine with stem cell platform to use it for the drug discovery or precision medicine. So I will first show you a movie that basically give you some brief introduction about the mechanism of CRISPR-based gene editing.
The CRISPR-Cas9 system is a tool for cutting DNA at a specifically targeted location. The technique has already revolutionized gene editing, but scientists are always looking for new possibilities. So what else can CRISPR do? Since being discovered in a bacterial immune system, CRISPR-Cas9 has been adapted into a powerful tool for genomic research. There are two components to the system: a DNA cutting protein called Cas9 and an RNA molecule known as the guide RNA. Bound together, they form a complex that can identify and cut specific sections of DNA. First, Cas9 has to locate and bind to a common sequence in the genome called a PAM. Once the PAM is bound, the guide RNA unwinds part of the double helix. The RNA strand is designed to match and bind a particular sequence in the DNA. Once it's found the correct sequence, Cas9 can cut the DNA. Its two nuclease domains each make a nick, leading to a double-strand break. Although the cell will try to repair this break, the fixing process is error-prone and often inadvertently introduces mutations that disable the gene. This makes CRISPR a great tool for knocking out specific genes. But making double-strand breaks isn't all CRISPR can do. Some researchers are deactivating one or both of Cas9's cutting domains and fusing new enzymes onto the protein. Cas9 can then be used to transport those enzymes to a specific DNA sequence. In one example, Cas9 is fused to an enzyme, a deaminase, which mutates specific DNA bases, eventually replacing cytidine with thymidine. This kind of precise gene editing means you could turn a disease-causing mutation into a healthy version of the gene, or introduce a stop codon at a specific place. But it's not all about gene editing. Several labs have been working on ways to use CRISPR to promote gene transcription. They do this by deactivating Cas9 completely, so it can no longer cut DNA. Instead, transcriptional activators are added to the Cas9 by either fusing them directly or via a string of peptides. Alternatively, the activators can be recruited to the guide RNA instead. These activators recruit the cell's transcription machinery, bringing RNA polymerase and other factors to the target, and increasing transcription of that gene. The same principle applies to gene silencing. A CRAB domain fused to the Cas9 inactivates transcription by recruiting more factors that physically block the gene. A more outside-the-box idea for using CRISPR is to attach fluorescent proteins to the complex, so you can see where particular DNA sequences are found in the cell. This could be useful for things like visualizing the 3D architecture of the genome, or to paint an entire chromosome and follow its position in the nucleus. CRISPR has already changed the face of research, but these new ideas show that what's been achieved so far could just be the tip of the iceberg when it comes to CRISPR's potential. Whatever comes next, it seems the CRISPR revolution is far from over.
So basically, CRISPR-based gene editing approach, there are two components in the CRISPR-based gene editing. So CRISPR, first they have an enzyme called Cas9, and then it's combined with a guide RNA. The guide RNA will basically help to recognize the location in the genomic DNA, and then basically drive Cas9 to the place that we want to edit it. And what Cas9 will do is Cas9 will basically cut the genomic DNA, and then the DNA start to start the self-repair. But the problem is there are errors happen during the repairing process. So then some errors will cause the frame shifting mutation of the genomic DNA, and then they knock out the gene and lost the function. And another type of editing is that people can deactivate the Cas9, so the guide RNA can still drive the deactivated Cas to the editing position, but this time they can add some other enzyme that can change the one single nucleotide. We call it precise gene editing that can cause the mutation in the cell types. So as I said, there are mainly two types of gene editing approach. One is to knock out the gene, and the other one is to induce the mutation, or correct a mutation. When people talk about CRISPR and stem cells, I mean, a lot of application, a lot of emphasis go to cell replacement therapy. Basically, we take patient samples, and then we reprogram into propotency stage. We call it induce pluripotent stem cells, and then we can use CRISPR to precisely correct the mutation in these pluripotent stem cells, and now we get these correct cells. They have the healthy or normal genomic DNA, and then the cells can be directed to a certain tissue or organs and put it back into patient for cell replacement therapy. But this is one application, but in today's talk, I will talk about another application. If we combine the CRISPR technology and stem cell technology, we actually have unlimited of cells that we can use for drug discovery or precision medicine. So what is precision medicine? This is an idea brought up several years ago, and it was also back to 2015. It's proposed by President Obama. So the idea is that if we know the genomic information of the patient, then the doctor can suggest some drugs, personalized drugs, based on their genomic information. So as these personalized drugs are supposed to be more efficient and less toxic. So there's a lot of interest on that in the last several years. There's a lot, both in academia and the industry, a lot of effort has been made on the sequencing part. And now we have a huge database with all this genomic sequencing data. But a little bit unbalanced part in this precision medicine is the drug discovery. If we check the FDA-approved drug library, very limited number of drugs are designed to target specific mutation or specific genes. Why is that? It's just some historical reasons. So if we go to the genome-wide association study, a lot of gene and mutations are associated with certain diseases. But without a very efficient and precise gene editing approach, it's very difficult to study the single mutation and understand their biological function. So let's come out to the second application of this technology. So now we can, again, take patient samples and put it back into induced pluripotent stem cells. And we can do CRISPR-based correction. So we call it isogenic line. And these two lines, most of their genomic information are identical. The only difference is one single mutation or a loss of one single gene. And now we can differentiate this pair of cells into the tissue organs we are interested in. And we can see what kind of cellular defect they have. And we can even run a drug screening and to find some small molecules that can particularly correct this defect. So in addition to correction of the patient-specific pluripotent stem cells, we can also use the healthy cells, and we can knock out a gene, or even we can induce a disease causal mutation. So that's another way we can study the function of biological function of one gene or one mutation. So now I will show you two examples that we use it to identify some drug candidate that can target some specific gene and mutations in disease progression. So the first story is about diabetes. So in this scenario, the GeneVault study has identified around 100 genes associated with type 1, type 2 diabetes. But for most of the genes, we really don't know their biological function. So we start from a human pluripotent stem cells, and we knock out the gene called CDKL1, and we make isogenic diabetes iPS cell, embryonic stem cells, and we differentiate them in pancreatic beta cells. Now we kind of monitor their biologic function. The major function of pancreatic beta cell is to respond to glucose stimulation. In the wild type cell, they can get around twofold induction. But in the knockout cells, they basically lose the response to glucose stimulation. So there's one defect we saw. And the other one is we also culture these cells in some diabetic-like conditions. We challenge them with high glucose, with high lipid condition. And we find that actually interestingly, CDKL1 knockout cells, they cannot really handle the stress very well. So they basically die more in the disease condition. So the interesting part is in regular culture condition, we don't really see any obvious defect. It's more related with type 2 diabetes. So then we run a drug screening. We screen around 2,000 compounds. This is our primary screening data. And we find one small molecule that actually can correct this defect. And when we treat the cell with this small molecule, we can see it brings the cell apoptotic rate back to the level comparable to wild type cells. It also brings the cell function back to the level compared to wild type cell. The interesting part is drug doesn't really have any effect on the wild type cells, which highlight the significance is specific for this particular gene CDKL1. Now we are making a panel of iPS cell lines, have a different diabetes gene and different diabetes associated mutations, and to see what the mechanism of the genes and whether we can find the different drugs to correct them. And what else can we do? Another model we are trying to establish, we call is humanized mice for drug testing. In the drug discovery field, more than 90% of drug people identified ex vivo actually doesn't work in vivo. So that's the reason we need animal models. We can use small animal models, mouse or rats. We can use large animal models, monkey or pig. But animals are not human. So some drugs, particularly some diabetes drugs, even though they work very well in the animal models, but they still fail in clinical trial. So what we're trying to do is now we still start from pluripotent stem cells. We use Cas to knock out the diabetes gene, and we make beta cells. And now we actually transplant into mice. And we can use some drug can specifically kill the mouse beta cells. Now the mouse are carrying human beta cells. They are totally dependent on the human beta cells to maintain their blood glucose levels. So then we can monitor their function. We can see the insulin secretion. If we transplant the wild-type cells, so the cell can respond very well to glucose stimulation. But for the knockout cells, they cannot respond very well. But if we treat these mice with the drug for around 4 weeks, actually they regain the ability to respond to glucose stimulation. And the cell we transplanted, we find it can survive very well for up to 1 year in the mouse model. So we can use this humanized mice model to perform the long-term testing for the efficacy and the toxicity. So in addition to the 2D culture humanized mice model, we are also interested in organoid model. So this is an example we use to study colorectal cancer. And colorectal cancer is a very aggressive disease, and they have an inherent form. It's basically if patient carry mutation of APC gene, most of patients in this family, they develop colorectal cancer around 20 years old, and there is really no treatment to this patient other than surgery. So they call family FAP patients. So we get fibroblasts from these patients, and we differentiate into induced pluripotent stem cells. And then we make 3D organoids as like pluripotent stem cell derived organoids. And we basically monitor their cell proliferations. We find that in the colorectal cancer patient derived organoids, the cell proliferate much faster than the wild type organoids. And then we actually perform a drug screening. It's a relatively small scale drug screening. And we find one small molecule is actually can rescue the overproliferation of the colorectal cancer iPSC-derived organoids to the level that are comparable to wild-type cells. And it's very important that in the same condition, the drug doesn't really cause any defect in the wild-type organoids, because cytotoxicity is a major side effect of most of chemotherapy drugs. So today I just show you 2 examples. We can use CRISPR-based gene editing to make isogenic human pluripotent stem cells, and we can use it to differentiate into 2D to different type to the cell type we're interested in. We can put it in put it in mice to make like humanized mice model. And then the 2D cells can be used for drug screening, and the humanized mice model can be used for the drug evaluation in vivo. And we also can get organoids that we can use for drug testing and small pallet drug screening. And we are trying to adapt it to more higher throughput now. What are missing? So I think we still need to do better job on the gene targeting part. Right now, knockout gene is very, very efficient, but knocking single SNPs is not good enough. Another thing is we want less off-target gene targeting approach, because if it targets several sites, it definitely will be a problem. And another thing we are working on that is, I think is very important, is for this disease modeling, we have to understand how to differentiate the stem cells into the cell types or organoids we are interested in and make sure they are functional. Yeah, that's it. Thank you. So I will pass it to Rusty.
Thank you, Shuibing.
Yeah.
I'm interested in neurogenesis. That's how cells emerge from a primitive state, it's like a stem cell, through an intermediate neuroprogenitor cell, and then how they differentiate into more mature cells. And we've done this a lot in mouse, and we've done this in monolayers, moving to human in monolayers using this IPS technology that we've heard about. But more recently, many of us are moving toward this three-dimensional structure with hopes that the organoids will provide a more realistic insight into the function of the human brain. Here we go. So this is basically the point I was making here. So we learned a lot about biology was disease and normal maturation using mouse models. And these are the 2D iPS cells, which I think both of these models are still very valuable and things that we learn a lot from. In particular, while the disadvantage of having a monocell or a couple cells in the dish is that they're unrealistic, the advantage is that we can really focus in on that cell's function and watch it in great detail. The recent advances in three-dimensional structure of cells allows us to do this 3D culture. Now, I put all three of them up here because none of them are replacing each other. They're all working cooperatively, and depending on the question that you're asking. But in this session, we're going to focus on the advantages of the organoids. The organoids are newer, and they're coming along, and a lot more work is needed to improve them. So they have some disadvantages right now, or let's say problems that need to be resolved. So let's see, yeah. So here's an example of the normal human cortical structure. This is sort of from a ventricular zone out to the outer edges of the cortex, and this is the mouse. And that's why you can see that it's important if we really want to understand things like cortical development, cortical organization, we need to use the human because it's much more complex than the mouse. These are both in vivo settings. So far, our human organoids are a good representation of a very early form of cortical development, but they don't give us a full maturation. And it's partly, as Alysson said, that the survival of the cells in this 3D structure is limited. But nevertheless, one can see these three-dimensional structures forming where you have this ventricular-like zone here with the primitive cells, and then on the outer edges, the more mature cells are formed in map 2. So one of the problems that we encountered when we were working with organoids initially was that they grow fairly large, and what one begins to see in the core of the organoids, at least the cerebral organoids, is there's a necrotic core that occurs if you allow them to get too big. If you keep them small by cutting them periodically, and you can get access to the nutriments to the core of them, but otherwise they have a problem in some cases. But also, as the other speakers alluded to, there's a significant amount of variability, so it doesn't happen in all cases. But it's something that we wanted to address. Now, the reasons for this, we believe, are that they are not vascularized, so the nutriments are not getting into the core of it. So how do we do that? Now, I should say that there are engineering institutes all over the world that are putting enormous amounts of effort to build a synthetic vascular system or ventricular system inside the core of this organoid. And I have to admit that we're not qualified as bioengineers to do that sort of work, and I suspect this success will come from that area. But we decided to take another approach at trying to get some vascularization. And it harks back to an old biological study that I had performed back about 35 years ago, where we were using in this days, the rodent brain as an incubator for human cellular tissues. They weren't iPS cells or organoids, but chunks of tissue that we wanted to get them survive and vascularized. And so a few years ago, together with a postdoc in the lab, we began to consider the application of this in vivo setting as a way to maintain the survival, help the survival of these cells. Now, what we use is a standard procedure for making organoids that Madeline Lancaster and Knoblish used, but we can use other methods for making different types of organoids that have different types of cells. We label all the human organoids in green so we can visualize them quite easily, and we mature the cells in culture for about 50 days so that they form a nice sphere, and then we implant them into this cavity that's made. Now, the unique features of this cavity are that they have a vascular bed overlying the top of a structure called the superior colliculus, and that is, when it's in its undamaged status,
the vasculature is a rich source of nutriments. And in the past, we had seen that vascularization can emerge into the human tissue. We wanted to see if that would occur in our tissue as well. Now, another feature of this that's useful is that in vivo, we have an imaging system that allows us to use high-resolution microscopy and watch the organoid in vivo over time as it matures. And since we've labeled all the human cells green, we can distinctly visualize them independently of the mouse. So this gives us an opportunity to look at live imaging. So here you can see an organoid at 1 day after implantation, and already by 10 days you can see blood surface on the surface of it as we look down on it from the top. Immediately, as we started looking at these cells, we began to see markers for CD31, which is an indicator of a vascular endothelial type cell, and we saw structures inside the organoid indicating that there was some sort of vascularization that was taking place, even as early as 14 days post-implantation. That's what DPI stands for. So to determine whether or not they actually vascularized the cells, we injected a rhodamine dye, dextran beads, into the blood system of the rodent, and then we visualized using this deep microscopy into the organoid, and we watched. We could visualize in the organoid and see blood vessels that had encapsulated the red dye, showing that the organoid had been vascularized. An important feature of this is that you can see these green cells on the surface of the vasculature forming the blood-brain barrier, or forming the surface around it, and these are human cells by virtue of the fact that they're green. Go back again. So it suggests that not only is it vascularized, but what we call a blood-brain barrier is formed because the dye is not leaking out into the surface. So we can now supply nutriments to the inner core of this system. Now, in addition to being able to demonstrate they're vascularized, we can also use this same imaging system to visualize the functionality of these cells. So here, 180 days after the post-implantation, so they're about 150 days now, we can label the cells with a calcium dye that is targeting mature neurons and monitor the activity of these cells. And here you can see that the neurons within the organoid are spiking in synchrony, suggesting that they're interconnected with each other, so some level of maturation is occurring. The usefulness of this is that we can actually come back to the same location in the rodent brain imaging of this human tissue over time, so we can monitor the changes that occur in those same circuits that are there. So the summary of this is we believe that we can say that these human cerebral organoids are amenable to transplantation in the rodent brain. They survive for long periods of time. We've looked at them for at least a year out after implantation, so it's a year plus 50 days. They become vascularized, and it looks like a blood-brain barrier is formed, so we can achieve nutriments in there. And they're functionally integrated. I would say that I didn't show you today, but not only is there evidence for innervation of the neurons with each other within the human organoid, but we have evidence that there's functional synaptic connections between the organoid and the host and the organoid that we're exploiting a little bit currently. So, thank you very much.
All right. Rusty, what time do you have to run?
I was going to say 12:30, so about 9 minutes.
Okay. So I think if anybody has questions, we should probably start with Rusty. And if not, I will start kind of a discussion. So one thing that, and this is for you, Rusty, I didn't realize in the beginning is that the blood vessels are not coming from the mouse, but they are being created inside the organoids.
Yeah.
So it means that there are endothelial cells inside the organoids. So when you start making them in transplant, they already have the different germline structures in there. Is that right?
Yeah, these are so actually in 2004 we showed that neural progenitor cells can, under certain stress conditions, generate endothelial cells. So that's part of the story that emerges here.
Right.
I wouldn't say that, while it does look very green, we're doing ultrastructure right now, looking at this in a lot more detail. And I would not be surprised if these are chimera, that there is some migration from the mouse in there, even though it looks very dark green.
Right, yeah.
But there's probably some chimera. We see this also in tumor formation sometimes, where the glioblastomas can form their own, they can contribute to their own endothelial vascularization. But if you look closely, you see some of the host migration into there as well. So it is a bit of a chimera. I don't know how much yet, but we're, actually one of the things that's fun about this is we can watch the emergence of angiogenesis. We can watch how blood-brain barriers are formed. And so that's one of the sort of the basic things that I think this has value for, is that there are very few cases where we can look at human brain vascularization forming, and we're hopeful that this is there. In a little more detail, not only do you have these large veins, but there's also all the microvasculature is pretty apparent too, and they're also forming blood-brain barrier. The EM, I think, is going to be really important to say what are the constituents, what are the other cell types that are contributing to it.
Another follow-up question. If you now transplant these cells into the kidney capsule or other regions in the animals, would you see the same thing, or you need kind of a brain environment?
Yeah, I really don't know, but I remember 10, 15 years ago when we first showed that the neural progenitor cells could give rise to endothelial cells. At that same time, others were showing that other stem cells could give rise to endothelial cells of the organ that they were transplanted in. So it looks like the local environment, and they have different properties in the different organs that they are transplanted to. So I haven't done that experiment, but it would be interesting to see, and then isolate the endothelial cells that are made, say, in the kidney capsule or some other organ, and see if they have features that are unique to the organ that they're transplanted.
Okay, good. I'll go to Matthias. Matthias, one aspect that I think it's fascinating, it is this idea of a tissue in a chip, right? And you heard like talks about brain organoids, and you are doing like the gut organoid. And there are several conditions now that are becoming apparent where there is a gut-brain axis. So do you think we can recapitulate that in a chip?
These are early days. So one of the challenges is to find conditions where we can grow these cells, happily inside a micro device. That's turned out to be more difficult than the field believed. And one challenge is that the materials that are used, to a large extent, are kind of very synthetic. So they're like polymers that have elastomeric properties, which is very useful to make chips, but for the cells to grow in them and start to self-organize has been quite a bit of a challenge. So I think one interesting avenue there is to replace in these micro devices these traditional materials with completely biological environments, with soft materials that are used to grow organoids conventionally. So this merging of sort of the biological world with the synthetic world has to be done in the coming years to make something as complex as you suggest. But I think eventually we'll get there. But there's a number of technical and biological challenges. And also, once you start to combine different tissues with each other, one other challenge is what's the right media to grow these different cell types or organ-specific culture conditions. That at the moment isn't something that's quite hard to do. It's a lot of experimentation to identify suitable media to grow different tissues in the same compartment.
And it's still, the brain-gut X communication doesn't happen directly, but through signals, right?
Long-range signals.
And also like from signals coming from the microbiome. And I understand that one of the goals of your research is really to populate these mini guts with different types of microbiome. Is that going on in your lab?
Exactly. That's one area where we're very excited about, is to try to open these cystic organoids that are very hard to access. The lumen, this is where the action takes place, where the the bacteria are growing, to open them up so that they can become easily accessible, and we can you know, perturb them in much more controlled manner. So what we're doing in the lab is to turn a cystic organoid structure into one that's a tube, which is open on both ends. So you can connect it to a fluidic system, to colonize it with bacteria, to remove them, to perfuse drugs, etc. So everything becomes experimentally much more tractable, and we have, I think, made some important progress along those lines.
Yeah, that's really cool. And going to the precision medicine aspect, right? I mean, it's fascinating that you can do all the precision medicine in vitro and find drugs that might compensate the mutations, right? But I think there is also some expectations that one day these would be doing in vivo as well. So how far are we from using genome editing in vivo? What's your take on that?
Yeah, I think for the in vivo part, definitely it's a huge group in the CRISPR-based gene editing part. People are working on that. And I think the first thing is efficiency, right? It really depends on the cell type people want to target. Because for us, we can now work on the Petri dish and the cell culture and 2D culture is always easier. And for your system, in 3D culture is already more complicated, but if in the whole human body, and it's efficiency and specificity, I think that's the two major issues people need to solve to do the in vivo-based gene targeting.
And talking about specificity in your experiments, I mean, how do you control for off-targets? How do you validate what you had is really because of that specific mutation and not something else?
Yes, so the ideal case, you can do the whole genome sequencing to validate there's no off-target effect, but practically it's just too expensive to do all of them. So we usually use two approaches to decrease the off-target effect. One is we always have several clones from each gene knockout or SNP lines. So in that scenario, the chance to have off-target effect will be a little bit. If they have different off-target effect, they may have different. They may target different locations. But if we see the same phenotype from multiple lines, most likely it come from our major target. And the second way we usually do is we on the CRISPR website you can actually predict the high risk of target effect. And sometimes we kind of sequence the top 10 or maybe top 20 to make sure the most risky areas, they are fine. Yeah.
Okay. We have a question.
I just had a question about the organoid technology specifically. It's fairly new, it's pretty new, and so I'm wondering if there are any established processes or procedures or media that are used on a regular basis. In other words, is there anything that is now making the organoid development a uniform thing so that it can actually be standardized?
Yeah.
I think it's a key point for the field to work on standardized materials. And I think there are a number of companies who are very active in this field and have commercialized media and entire kits to grow organoids. And this seems to work quite well as far as I heard from people who are using it. And I think it's an important step to make organoid technology widely accessible and really fit for real-life application in industry and so forth.
But I'll add on that as well, because, I mean, as you pointed out, it's such a young field that in one side, you want to create something more robust, reproducible among labs. On the other side, you don't want to block the creativity. I mean, this new protocol that we had was almost like by chance, people kind of empirically testing other conditions and end up gaining functional sophistication in the networks by media optimization or changing some factors. So it goes in both directions, right? I mean, you want more reproducibility, but you also want to explore more. I think it really depends on the use of the technology and where you are in your lab. If you are, I mean, pushing forward to have better models, or if you are happy with the model you have, in understanding the limitations, and if it's useful for the question you're trying to answer.
And is it the case with CRISPR-Cas9 that there are already ways that that's done, so everyone has a standard way of using that technology?
I think even for Cas9 field, we have some standard technology people use every day, but in the meanwhile, the field still move forward very fast, right? And they have kind of very precise gene editing now, and also like RNA-based gene editing. So in that scenario, there are always new technologies developing. I actually have a question for Matthias,
Okay.
because we're also interested in the organoids on-chip model, and you just mentioned, I think the trouble we had a lot is to find a common media to culture different organs together. But then we think back about that because different organs, they are controlled in different microenvironment, and in human, they are not really exposed to the same media, right? So they have the blood vessels contact it together. So practically, I know it's definitely more complicated, but that may be a way to go, so that in each organ they still have their own microenvironment, but you build something like a blood vessel or that artificial blood vessel.
It's the way to go.
Yeah.
I think blood is sort of the common shared pool of nutrients and growth factor, and that's the reason why FBS works so well, because it has all these things in it, so you can grow almost every type of cell. But you want to get rid of it because it's very complex and ill-defined. But I think that's the idea here, that we're engineers and many groups are trying to build a vasculature that can, be shared among multiple tissues. Yeah, I think that's exactly the approach.
I think the NIH has lots of initiatives to build better tissues in a chip by communicating to each other through bioengineer connections that mimics this idea of blood vessels. Might not be the blood vessels per se, but you're in touch. At least the signals can move from one tissue to another. I mean, this is really exciting.
There's one point I want to add, which I think is biologically and technically very interesting, is that right now when we build organoids, they're composed of largely one type of tissue. They lack a lot of components from the real organ. And I think once you start to add these other components, for instance, if you have an epithelium and you add a mesenchyme, the system becomes more autonomous because there's crosstalk between the mesenchyme and the epithelium. And maybe that makes life actually easier once you then want to combine multiple organ models in one setting. You see? So I think the more complex an organoid becomes, the more autonomous it will probably be functioning. I think that's an interesting challenge.
Yeah. All right. Okay. So thank you for participating on this panel. It was a pleasure to hear about your science, and I think we end here.
Okay.